Bismillahirrahmanirrahim. Dear student, Assalamu alaikum. Today the topic of our discussion is high performance liquid chromatography. In our previous lecture, we discussed different types of chromatography. And you remember this was the last slide, and we discussed that how liquid chromatography changed into high performance liquid chromatography. Actually, in chromatography, high degree of separation was needed. For that thing, refinement of a packing material was needed. So the packing material size was reduced up to 3 micrometer, depends, sometimes 3 micrometer, 5 micrometer, and 10 micrometer. Also, to reduce the analysis time and delivery of this eluent by a pump was needed. So high pressure pump, and also a demand of a special equipment that can withstand this high pressure was needed. That's why this liquid chromatography, because of these changes, this liquid chromatography was changed into high performance liquid chromatography, which is called HPLC. So HPLC is a form of liquid chromatography used to separate compounds that are dissolved in a solution. Means basically this HPLC is an example of liquid chromatography. So HPLC instrument consists of reservoir of mobile phase, and this mobile phase is pumped by a high pressure pump, and then an injector which can inject this sample, and this sample along with mobile phase is passing through a separation column, and the different components which are eluting from the column are detected by detectors. So compounds are separated on the column in which sample mixture is passed through this column. The different component in the mixture pass through the column at different rates due to difference in their partition behavior, due to the uh, due to the interaction of these different components with the different phases. The mobile phase must be degazed before the column to eliminate the formation of air bubble in the column, because that can affect the column efficiency then. This is the flow chart of HPLC system. So in HPLC system, basically there are different solvent reservoir. So sometime 2, sometime 4, 3. So this is solvent 1 and solvent 2. So this solvent is degazed here before mixing. When these solvents are mixed, this is pumped by a high pressure pump to the column, but most of the time, or even sometime before the actual column, a pre-column is also there, which can stop all the unwanted things from the column, because column is an expensive thing in HPLC. Also, it can affect the efficiency of a HPLC system. So after the pre-column, the sample injection port, which is this sample is injected into the flow of this mobile phase, which then this mixture is passed through this column, analytical column, and then like here also this flow splitter, which can split the flow of the mobile phase as well as the sample. So some of the mobile phase, means a very small part, also where the sample is passed through the column, and most of these sample as well as this mobile phase is going into the waste, and then at last here these different components are detected by detectors, and then it give us a chromatogram in a computer. So basically, the chromatography of this HPLC system is here on the column, because this column material decide that how these different components are separated, how well these components are separated from each other. So what is the chromatography? So the we discussed different types of chromatography in our last lecture. So in this column, so there are also different types of chromatography. One is partition chromatography. We have discussed this partition chromatography in detail in our previous lecture. And one is adsorption, or called liquid solid chromatography, and ion exchange chromatography. So this column material is made of ion exchange resins, and size exclusions are gel chromatography. So this chromatography, either the material decide whether it is a partition chromatography, adsorption, or ion exchange, or size exclusion gel chromatography. This is typically HPLC system, which is lying there in our lab. So this is HPLC system connected to a computer. So we'll discuss one by one. First of all, these bottles you can see, these are the solvent reservoir. Different solvents are used: methanol, acetonitrile, sometimes buffer, etc. Different solvents are used. These are solvent reservoirs. And here, like, the solvent is coming from the solvent reservoir. Here in this region of this HPLC system, a solvent delivery system, high pressure pump is set there, which can deliver this solvent by a high pressure. And then the injector here in this system, like here in this region, injector, also degasser is also here in this system before the solvent going through the column. So here, injector which can inject the sample, and also a splitter which can split the sample and the solvent system. Some of this solvent and sample is going to the waste, and the rest here in this area, here the sample is there, sample is here. This is usually, this is an automatic system, which where this system can pick this solvent vial, and solvent is in sample, is a sample vial, sorry, sample vial, and where sample is injected into the part of this solvent, which is going here in this system. Here the column is fitted in that part of the chromatography, and this solvent along with sample is passing through the column, which are different components are separated. Here in this part of chromatography, detector, usually a diode array detector, is set there, and waste collector is also lying here, which can, the waste is coming from here to collect, and here, and then this computer system, this recorder or data collection system is there. So this is the typical HPLC system, consists of different components. So what are the uses of HPLC? This technique is used for in chemistry and in biochemistry research, analyzing complex mixture, separating different component of a mixture. It is used to purify different chemical compounds. Uh, Usually organic chemists do purify chemical compounds which they synthesize in their lab, developing processes for synthesizing chemical compounds, also isolating natural products, also the predicting the physical properties, also used in a quality control to ensure the purity of raw material, to control and improve the process yield, like in organic chemistry, to quantify assay of final products, or to evaluate the product stability, or even to monitor the different components during the degradation processes. In addition, it can used for analyzing the air and water pollutants, for monitoring materials, for the occupational safety and health processes, and for monitoring pesticide level in the environment. Different federal and state regulatory agencies use HPLC to survey food and drug product, for identifying confiscated narcotics or to check for adherence to label claims. So all these things, these are the major uses of HPLC. First of all, the HPLC chromatograph injectors. The function of the injector is to place the sample into the high pressure flow in a as narrow volume is possible so that the sample enters the column as a homogeneous low volume plug to minimize spreading of the injected volume during transport to the column. The shortest possible length of tubing should be used from the injector to the column so that the flow is not affected. So when injection is started, an air actuator rotates the valve, solvent goes directly to the column, and the injector needle is connected to the syringe. The air pressure left the needle, and the vial is moved into the position beneath the needle. Then the needle is lowered to the vial. HPLC column is called the heart of the column, HPLC system. Column is one of the most important component of the HPLC chromatograph because all the separation you need is done there on the column. All the sample components is achieved when these components pass through the column. The high performance liquid chromatography apparatus is made out of stainless steel tubes with a diameter of 3 to 5 millimeter and a length ranging from 10 to 30 centimeter. Normally, columns are filled with silica gel because its particle shape, surface properties, and pore structure help to get a good separation. Silica is wetted by nearly every potential mobile phase, is inert to the most compound, and has a high surface activity, which can be modified easily with water and other agents. Silica can be used to separate a wide variety of chemical compounds. Its chromatographic behavior is generally predictable and reproducible. These two are the very important factors. That the behavior of silica is predictable and reproducible. This is the picture of an HPLC column, that how HPLC column look like. Is I mentioned it, that this column is made of a stainless steel. Inside this stainless steel, usually the column material is packed in a small and a very compact size. Usually that is silica. Some even polymeric bone columns are also there. This is picture of an HPLC column. So question is, what affects system basically? So the column parameters are column material, that what type of column material, the deactivation of this column material, stationary phase, what is stationary phase of this, and coating material. All these things can affect the system. They affect the separation efficiency, and also these are the instrument parameter. So in a whole, column parameter and instrument parameter can affect the separation efficiency. So temperature, usually like at a higher temperature, the results are different. The peak shape is different, and flow of the mobile phase, and also signals and sample sensitivity, how this column is this system is sensitive to this sample, and a detector, which type of detector you are using. These are these column parameter, instrument parameter can affect the system, and sample parameter are also can affect the system, like concentration. What is the concentration of the sample? What is the matrix? How much matrix is there? So that can affect also a system, and also solvent effect. Which type of solvent you are using? Uh, Methanol or acetonitrile, or which buffer you are using, what is the pH of the buffer. So, and also sample can affect the system. So columns are of different types. Several types of column can be used, uh, and even on the basis of this column, we called it normal phase chromatography or normal phase column, and other is reverse phase column, and size exclusion column, and ion exchange column. So, what is normal phase or normal phase chromatography or normal phase column? In this column type, the retention is governed by the interaction of polar part of the stationary phase and solute. For retention to occur in a normal phase, the packing must be more polar than the mobile phase with respect to the sample. Because of this, because this is usual thing that the for retention to occur now, the packing material must be more polar than mobile phase, and exactly the opposite of this normal phase chromatography is the reverse phase chromatography, like next one, this one. So in this column, the packing material is relatively low polar or non-polar, and the solvent is more polar with respect to the sample. So retention is the result of interaction of non-polar component of the solute and the non-polar stationary phase. Typical stationary phases are non-polar hydrocarbon, waxy liquids, or bonded hydrocarbons such as C18, C8, and the solvent are polar aqueous organic mixtures such as methanol water or acetylnitrile water. Some buffers are this. So size exclusion. In this size exclusion column, the HPLC column is consist of substances which have controlled pore size and is able to be filtered in an ordinary phase according to its molecular size. Small molecule penetrate into the pores within the packing, while larger molecule only partially penetrate the pore. The larger molecule elute before the smaller molecule in this size exclusion column. The last one is this ion exchange column. In this column type, the sample components are separated based upon the attractive ionic forces between molecule carrying charge group of opposite charge to those charges on the stationary phase. So if the surface of the stationary phase is negative, your sample components which you are separating must be positively charged so that interaction are created there, interactions are happening there. Separations are made between a polar mobile liquid, usually water containing salt or small amount of alcohol, and a stationary phase containing either acidic or basic fixed site. One of the most important thing in chromatography is selectivity factor. K value tell us where bands elute relative to the wide volume. Where band eludes relative to the wide volume, these values are unaffected by such variables as flow rate and column dimension. The value tell us where two peaks elute relative to each other. This is referred to as selectivity factor or separation factor. So there are different types of detector in HPLC system: absorbance, UV filter, UV with monochromator, also IR absorbance detector, fluorescence detector, refractive index detector, evaporative light scattering detector, electrochemical detector, mass spectrometric detector, photodiode array detector. These are the different types of detector used in HPLC chromatography. So you can check at least you should know the basic of these detectors, that how it works basically. So the evaluation parameters of chromatographic system are the efficiency of the column is that how much sample was loaded and how much you get. This is called basically the efficiency or recovery, and resolution, that how two peaks are well separated from each other. This is the resolution, and inertness, how your stationary, how this your mobile phase and all these things are inert towards the sample components, should not react with that. And a retention index, how much it retain the different components, and also column bleed, some of the column bleeding. So you should know this, what is this column bleeding, and capacity factor, as I mentioned it now. So this was all about the chromatographic system, that how chromatographic system HPLC work. These are the two books that you can use for this HPLC topic. One is this J.T. Christian Analytical Chemistry 7th edition, well-written book, and Scooge and West, Fundamental of Analytical Chemistry 9th edition. So this was all about the HPLC, not in very much detail is will be in our future, in our next semester or something. But hopefully we will discuss next time the gas chromatography in detail. Thank you very much.